Hello students, welcome to classroom teaching. In this session, we are going to discuss about 3 points only. The first is apparent size of an object. The second point that we are going to discuss, it is uses of concave lenses. And the last one is persistence of vision. So let us start with first point, that is apparent size of an object. Student, you first see a small clip in this video and you can understand what I am going to tell about apparent size of an object. In this small clip you can see that as the object is going away from us, its size appears to decrease. That means it is becoming smaller and smaller and smaller. So why it happens actually? Suppose you are looking from this and suppose the object is very close to your eye. Suppose this is an object. So now the object is very close to your eye. Then the angle made by this object with your eye, suppose it is alpha. If the object is going away from you, now the size of the object is same, because we are not changing that object. The object is same one, but it is now away from your eye. So the angle made by this object with your eye, now it becomes smaller, and it is, suppose it is beta. This is object PQ. So suppose the object is still, it goes away from you. So the angle it is made by this object with your eye, it is still becoming smaller. So suppose it is gamma. So as the object is going away from you, then the angle made by your eye with that object, or the angle made by that object with your eye, it is becoming smaller and smaller, and therefore the object appears to be smaller. So this is nothing but the apparent size. That is not actual size. Actual size is not changing. This is apparent size of an object, and it depends on the angle made by that object with your eye. Clear? So let us discuss about the second point that I told you, that is nothing but uses of concave lens. So let us see the uses of concave lenses. Actually, there are so many uses of concave lenses. In medical equipments, in scanner, in CDs, CD writers or CD players. Actually, in these instruments, laser beam is used, and to improve the quality and for proper functioning, proper working of these equipments, lenses, concave lenses are used there. Then in peephole camera means which are peephole which is fitted in the door. There also concave lens or more than one, that means two concave lenses are used to get wider view of the what is present outside the door, or to get the large view of what is present outside the door. So there also concave lens is used. Then the well-known use that we know, that is to cure myopia, that we learned in last session. So to cure the defect of myopia in spectacles, myopia or nearsightedness, so in spectacles concave lenses are used there. Then in torches, that means to get the sprayed beam of light, that means wherever we require the light rays to spread, at that time we get or we require concave lens or diverging lens, that we can say. Then in cameras or in microscopes or in telescopes, usually we use convex lens there, but to get better quality of image, concave lenses they are also used along with convex lenses in eyepiece. So whenever we use the convex lens as an eyepiece, along with that we use concave lenses to get better quality of image. So these are the uses of concave lenses. So let us discuss the third point that I say, that is nothing but persistence of vision. Before going directly to the persistence of vision, you just see a very small clip of just 10 seconds, and then we will discuss about this persistence of vision. In this clip you can see that the object, or there are so many pictures which are drawn there, so many drawings are there, and it appears that it is moving. So it is the effect which we call it as persistence of vision. Students, what happened actually, as soon as the object, or why we can see the object, we can see any object, I can see this marker here. Why? Or you can see this marker here, because the image of this object, it is formed on our retina. And as soon as I remove this object from your or from in front of your eye, as soon as I remove this object, the image which is formed on retina, it is also given out, or it also washes. It is also removed from the retina. But this process is not instantaneous. What is the meaning of that? That means though I remove this object from in front of your eye, the image of this object, it remains on the retina for 1/16 of second. 1/16 of the second. The impression or the sensation of that image or the persistence of the image, it is there on retina after removing the object. After removing the object for 1/16 of second, it appears to be on the same place. The impression of that image, it remains there on the retina, and within 1/16 second, if another image is formed in front of you, then we cannot, or our brain cannot perceive it, our brain cannot understand it. So all these images get mixed into one another, because the impression of the first image is already there, and within that period another image is formed on the retina, and therefore we can see it in continuation. A very small example is given in this book, you should try that activity. Or we can see this activity here also. Let us see that activity. So in this activity, this is nothing but essence stick. You can see it here, I place it like this. You can see it here, I place it like this. You can see it there, here. But now we can see the complete circles of the fire, because the image is formed, the new image is formed here within 1/16th of second. So in movies, the same technique is used. They take pictures, many pictures having slight difference, and all the pictures they run from in front of your eye within 1/16th of second, and due to that we can see the continuity in that pictures. Our brain cannot separate these pictures. So this is nothing but persistence of vision. Actually, we discussed earlier in last video, in last session, that there are two types of cells on our retina. That is cone cells and rod cells. So how our brain, it perceives different colors. So for that, cone cells are responsible, and rod cells, they are responsible for the brightness of the color of the or the brightness or dimness. As the rod cells, they work only in bright light. Therefore, in dark or wherever there is a very dim light, we cannot recognize colors. There are some problems in some persons. If they don't have that much cone cells or cone cells responsible for a particular color, then those persons, they are color blind. That means they cannot perceive colors, they cannot separate different colors. So we will stop here for this session, and in next session we take a new session, or we will take a very small session again for the uses of convex lenses, because there are so many uses of convex lenses which we take in detail. So thank you very much.